Hello, I'm Gretchen Colwell, and you are about to view my webinar on genetics and developmental disabilities. This presentation is a requirement of Westchester University's KIN 583, Medical and Pathological Issues Related to Disability. The objectives of this webinar are as follows. Know about the human genome and its implication for the origins of developmental disabilities. Be able to explain errors in mitosis and meiosis, including non-disjunction, translocation, and deletion. Know the differences and similarities among autosomal recessive, autosomal dominant, and X-linked genetic disorders. Understanding the concepts of genomic imprinting, anticipation, and mitochondrial inheritance. Understanding the ways in which environmental and heredity contribute to the development of multifactorial disorders. Before we get into looking at genetics and chromosomes more specifically, here's a brief Genetic 101 video to help jog your memory back to 10th grade bio, before I get into a more intricate discussion.
Your body is made up of 50 trillion cells. Cells come in many different varieties, with many different functions. But inside almost every cell is a nucleus, containing 99.9% of your genes, and mitochondria, containing a few more genes. All told, you have nearly 20,000 genes. Your genes are small parts of a long molecule called DNA, or deoxyribonucleic acid. If you lined up all of the DNA containing all of your genes, it would measure 6 feet long. But it's coiled so tightly that it fits in just one cell nucleus. DNA is a double-stranded molecule composed of sugar, phosphate, and 4 different bases: adenine, thymine, cytosine, and guanine. These bases spell out the language known as the genetic code. The number and order of these 4 bases determine, for example, whether you are a chimp, a cow, a banana, or a human. Most genes are recipes for making specific proteins. These recipes are passed down from parents to children, from generation to generation. When someone says, "You have your father's hair," what they mean is, you appear to have inherited a gene or genes from your father that makes a protein that instructs your hair follicle cells to produce hair that curls like your father's. But they usually opt for the shorter version. Genes tell a cell how to function and what traits to express. More specifically, gene regulators turn different genes on and off in different cells to control cell function. The long molecules of DNA containing your genes are organized into pieces called chromosomes. Different species have different numbers of chromosomes. Humans usually have 46 chromosomes, 2 sets of 23, or simply 23 pairs of chromosomes. Chimpanzees have 2 sets of 24, or 24 pairs of chromosomes. Rhesus monkeys have 21 pairs of chromosomes. Cows have 30 pairs of chromosomes. Chickens have 39 pairs. Fruit flies have 4 pairs. And bananas have 11 pairs. So, what percentage of the DNA in your chromosomes do you share with other species? You share 93% of your DNA with the rhesus monkey and 98.5% with our friend chimpanzee. How about with other humans? 99.5%. So, what makes us different from one another? Well, for one thing, SNPs.
I hope you were able to follow along with the video. It gives a nice basic understanding of what we're about to get into. Hopefully you can bear with me as we go through this presentation. There's a lot of complicated material. Just follow along and we will get through it together. So cells are divided into two compartments, the central core and the outer area, or the nucleus and the cytoplasm. The nucleus contains chromosomes, which contain genetic code, or DNA. The nucleus contains the blueprint for an individual's development. The cytoplasm manufactures the products needed to complete the task of development. Genes are responsible for our physical attributes and biological functions. Here's an overview of what the human cell looks like. It's a still photo. The video kind of passed through what this looks like, so take a moment, jog your memory. I'm sure you've learned this at some point throughout your schooling. So let's take a closer look at chromosomes. In humans, each cell contains 46 chromosomes. Each chromosome contains hundreds of genes, some more, some less. Chromosomes are organized into 23 pairs of complementary chromosomes, one from the mother, one from the father. When egg and sperm cells, or germ cells, meet at conception to produce a fertilized egg with the full complement of 46 chromosomes, X and Y. 22 chromosomes are termed autosomes. The 23rd pair consists of the X and Y chromosomes, or the sex chromosomes. Y determines maleness, so XX is a female and XY is a male. Cell division and its disorders. So let's look at the breakdown between mitosis and meiosis. Mitosis is non-reductive division. There are two daughter cells, each containing 46 chromosomes, that are formed from one parent cell. It occurs in all cells. Now, meiosis is a reductive division. 4 daughter cells, each containing only 23 chromosomes, are formed from one parent cell. This occurs only in germ cells and creates sperm and eggs. The ability of cells to continue to undergo mitosis throughout the lifespan is essential for proper bodily functioning. Cells divide at different rates, however, ranging from once every 10 hours for skin cells to once every year for liver cells. This is why a skin abrasion heals in a few days, but the liver may take a year to recover from hepatitis. By adulthood, some cells, including neurons and muscle cells, appear to have a significantly decreased ability to divide. This limits the body's capacity to recover after, say, a stroke or a sports injury. So as we get older, those basketball injuries take a little bit longer to heal. Here's a nice breakdown of crossovers. One of the primary differences between mitosis and meiosis can be seen during the first of the two meiotic divisions. During this phase, the corresponding chromosomes line up beside each other in pairs. Unlike in mitosis, however, they intertwine and may cross over, exchanging genetic material. Although this crossing over of the chromosomes may result in disorders, it also allows for the mutual transfer of genetic information, reducing the chance that siblings end up as exact copies of each other. Some of the variability among siblings can also be attributed to the random assortment of maternal and paternal chromosomes during the first of two meiotic divisions. Non-disjunction. This is the failure of a pair of chromosomes to separate during mitosis or meiosis, resulting in unequal number of chromosomes in the daughter cells. An example of this, the result is that one daughter egg or sperm contains 24 chromosomes and the other 22 chromosomes. Usually, these cells do not survive. Trisomy. This is a condition in which there are 3 copies of one chromosome rather than 2. Trisomy 16, or embryos with trisomy 16, are never carried to term. Trisomy 13, 18, and 21 are most commonly observed chromosome disorders at birth. Trisomy 16, reflecting back, the reason they believe they do not carry to term is that the chromosome 16 contains so many genes important to development that its disruption is incompatible with life. The other 3 probably survive because these chromosomes contain a relatively small number of gene loci, and their disruption is compatible with life. Even so, the majority of fetuses carrying chromosomal abnormalities are spontaneously aborted. Among those children who survive these genetic missteps, intellectual disability, unusual facial appearances, and various congenital malformations are common. Let's look at chromosomal gain. One example would be Down syndrome. This is the most frequent chromosomal abnormality is non-junction of autosomes. Most commonly occurs during meiosis. Both copies of chromosome 21 end up in one cell. The most common, like I said earlier, is Down syndrome or trisomy 21. When non-disjunction occurs during the first meiotic division, both copies of chromosome 21 end up in one cell. Instead of an equal distribution of chromosomes among the cells, 23 each, one daughter cell receives 24, where the other only receives 22. The cell containing 22 chromosomes is unable to survive. However, the egg or sperm with 24 chromosomes occasionally can survive. After fertilization with a sperm or egg containing 23 chromosomes, the resulting embryo contains 3 copies of chromosome 21, or trisomy 21, resulting in Down syndrome. Chromosomal loss. An example we'll look at is Turner syndrome. This affects girls. It's the only disorder in which a fetus can survive despite the loss of an entire chromosome. So monosomy, which is a chromosomal disorder in which one chromosome is absent. More than 99% of this incident is miscarried. It's affected by meiotic errors in sperm. There is no sex chromosome from the child's father. Turner syndrome continued. Children will have short stature, webbed neck, broad chest, widely spaced nipples, non-functional ovaries, 20% have obstruction of the left side of the heart, visual perception impairments, and typical intelligence. Human growth hormone injections have been effective in increasing height in girls with Turner syndrome, and estrogen supplementation can lead to the emergence of secondary sexual characteristics. However, these girls remain infertile. Mosaicism. This is the presence of two genetically distinct types of cells in one individual. Children with mosaicism often appear as if they have the condition, although physical abnormalities and cognitive impairments may be less severe. For example, a child with mosaic form of Down syndrome may have trisomy 21 in blood cells, but not in skin cells or in some, but not all brain cells. Translocations. This is the transfer of a fragment of one chromosome to another chromosome. An example of this is that a portion of chromosome 21 might attach itself to chromosome 14. If this occurs during meiosis, one daughter cell will have 23 chromosomes, but will have both a chromosome 21 and a chromosome 14/21 translocation. Deletions. This is the loss of genetic material from a chromosome. There are visual deletions and microdeletions, or contiguous gene syndrome. Kreiderschat syndrome is a form of visible deletion, and Williams syndrome and VCFS is a form of microdeletions. Visible and micro are determined by how geneticists would look at the genetic material. The frequency of chromosomal abnormalities are as follows. 25% of eggs and 3% of sperm. So they're missing or have an extra chromosome. 1% to 5% have a structural chromosomal abnormality. 10 to 15% of all conceptions have a chromosomal abnormality. 50% are trisomies, 20 are monosomies, 15% are triploids, Remaining are structural abnormalities and tetraploids. More than 95% of fetuses with chromosomal abnormalities do not survive to term. Genes and their disorders. Genetic disorders can also result from an abnormality in a single gene. The human genome contains approximately 20,000 to 25,000 genes. Genes are composed of various lengths of DNA that, together with intervening DNA sequences, form the chromosome, which is a double helix as we saw in the video. The nucleotide bases are the following four elements: cytosine, guanine, adenine, and thymine. The minimum number of genes necessary for life is approximately 300, and all living organisms share these 300 genes. The sides of the ladder of the double helix are composed of sugar and phosphate molecules, whereas the rungs are made up of the four chemicals which I listed previously. Genes range in size, containing from 1,500 to more than 2 million nucleotide base pairs. All genes are not turned on at all times. Some are only active during fetal stages, and some are never turned on, hopefully, such as cancer. This turning on and off plays a particular important role during fetal development. As a result, problems in the on-off switching can be devastating at this time. This is transcription. The process in which messenger RNA is formed from a DNA template. In all RNA, the nucleotides are the same as those in DNA, except that uracil substitutes for thymine. Errors may occur during transcription, causing problems in development. Translation. This is the process in which an amino acid sequence is assembled according to the pattern specified by messenger RNA. Once transcribed, the single strand of messenger RNA detaches and the double-stranded DNA zips back together. The messenger RNA then moves out of the nucleus into the cytoplasm, where it provides instructions for the production of a protein, and this is what we call translation. The following slide, we see this process happening. We take transcription and translation and show you the interaction with DNA and out into the cytoplasm, the proteins that are formed. Once in the cytoplasm, the messenger RNA attaches itself to a ribosome. The ribosomes move along the messenger RNA strand, reading the message of the three-letter codons, such as GCU, CUA, or UAG. Most of these triplets are codes for specific amino acids. Once the proteins are complete, the messenger RNA, ribosome, and protein separate, and the protein is released into the cytoplasm. If not used in the cytoplasm, the protein is sent to the Golgi apparatus to be packaged and released throughout the body. Mutations. A change in the gene that occurs simply by chance. So first we'll look at point mutations. These are the most common type of mutations. No adverse consequences, and may cause a nonsense mutation. A nonsense mutation results in a change in the triplet code that substitutes a different amino acid in the protein chain. Essentially, in the nonsense mutation, a stop codon prematurely terminates the protein formation. Examples of where this has occurred include neurofibromin, which leads to a high incidence of ADHD. Insertions and deletions. These are the most common mutation in individuals with spinal muscular atrophy. Involves an insertion in the survival motor neuron gene, rendering it defective. The most common mutation in Duchenne muscular dystrophy involves deletion. Additions or subtractions, or insertions and deletions, may also lead to a frame shift in which the 3 base pair reading frame is shifted. All subsequent triplets are misread, often leading to the production of a stop codon and non-functional protein. An example of this mutation can be seen in Tay-Sachs disease. Triplet repeat expansion. In some nucleotide triplets, they repeat multiple times in certain regions of normal genes. If the number of triplet copies is expanded markedly, a gene can be turned off, leading to diseases such as Huntington's and Fragile X syndrome. Selective advantage. This is a very interesting concept, and we will be watching a video momentarily. Natural selection. This is the process of adapting the environment. The process, according to Darwin, by which organisms best suited to survival in their environment achieve greater reproductive success, thereby passing advantageous genetic characteristics on to future generations. With regard to selective advantage, we're going to watch this clip on how sickle cell anemia gene would be considered a disadvantage in most minds, but it has become part of selective advantage with regard to malaria.
For thousands of years, coastal West Africans have fished the sea to make a living. But 4,000 years ago, a deadly disease arrived here, bringing death and misery. Its name, malaria. That people continue to live and fish here today demonstrates the power of evolution. Power felt in the widespread effect of just one mutation in a single gene. Malaria is caused by a tiny parasite carried by mosquitoes. Humans become infected with the parasite through a mosquito's bite. It may take just a few short weeks for the parasite to destroy a human's red blood cells, necessary for spreading oxygen throughout the body. A red blood cell is invaded by the parasite, which soon multiplies, causing the cell to burst. These new parasites are now free to invade yet more cells. In West Africa, malaria is so common that most children are infected with the disease. Though many suffer symptoms severe enough to warrant trips to the hospital, like this one in Senegal, for most the disease is not fatal. The key to their resistance is in their genes. Dr. Ronald Nagel studies the genes that make hemoglobin an essential part of red blood cells. Genes are all paired, with each parent supplying one half of each pair. If either hemoglobin gene undergoes a mutation, the hemoglobin it makes will be changed. This particular mutation, called the sickle cell gene, is tiny, but it's enough to change the shape of the hemoglobin molecules it makes.
People that had one copy of the gene were able to survive their infection, the malaria infection. They would not die of it. Hence, they were able to grow up, get married, have children, and pass the genes on to the next generation. This is selective pressure. That gene had an advantage in that particular environment for those carrying it.
We all have lots of small gene mutations. Mostly they go unnoticed. But if the environment changes, one of them may suddenly show unforeseen effects, good or bad. In this case, one copy of the gene is beneficial, but two can be disastrous, as Fatu and Sheikh Assar found when they started a family. Both Fatu and Sheikh are protected from malaria because they each have a single copy of the sickle gene. Their children each had a 50% chance of receiving one single copy of the mutated gene, or 1 in 4 might get both parents' normal genes, leaving that child unprotected. Another 1 in 4 might inherit both sickle genes, and that is the case for their daughter Fatu, named for her mother. Because she inherited both copies of the sickle cell gene, all of Fatu's hemoglobin is adversely affected, not just half. Her molecules turn into rigid rods that provoke sickling of the red blood cells, even in the absence of malarial parasites. The slightest exertion can cause a sickling emergency. Her cells sickle one after another, causing blockages in her narrow blood vessels. Fatu's blood flow is cut off, and she experiences agonizing pain. The sickling caused by the mutation led scientists to name this disease sickle cell anemia. Fatu's life will be far from comfortable, but medicine can help her survive this debilitating disease. Though the sickle defect was a mistake, causing great harm to some, for others it is priceless protection against malaria. This is how evolution works. Mutations occur at random and may or may not have positive consequences.
The more diverse people are, the more likely that population is going to survive, even to things that might come in the future, infections that we don't know about now. Some people might be, by chance, more resistant to that future infection that might wipe out a population. If everybody's the same, if everybody has the same genetic background, a population can be easily wiped out by a single plague.
Okay, I hope that helped you better understand natural selection and how selective advantage works. Next, we look at single nucleotide polymorphisms, which is a big word for the variations in DNA sequence. Of the more than 3 billion base pair genetic codes, people of all races and geography share 99.9% genetic identity. Although this is quite remarkable, that 100th% difference means there are about 3 million DNA sequence variations. This is the basis of evolution that can contribute to health, unique traits, and disease. Now we look at single gene disorders, or Mendelian. Okay. And the 3 variations here are autosomal recessive disorders, autosomal dominant disorders, and X-linked disorders. But first I'm going to show another clip so you can understand how Mendelian discovered this phenomenon.
Hi, it's Mr. Anderson, and welcome to Biology Essentials video 29. This is on Mendelian genetics, and it's called Mendelian genetics. It's named after Gregor Mendel. In biology, there are 2 famous names: Darwin, who was famous and controversial in his own time, and Gregor Mendel, who died in obscurity. But both of them made huge advances in the field of biology, and Gregor Mendel in the area of genetics. If they could have gotten together, those 2 theories, when they finally came together as modern synthesis, it was really powerful. Unfortunately, he dies in obscurity. But this is what he did. He was crossing pea plants, and so he can take one pea plant and you use a paintbrush to transfer pollen from one to another so you know who are the parents. He would then create offspring as a result of that. Now, peas are great because they have a number of different characteristics, and more importantly, you can make a lot of them really quickly. So in a pod, each of these peas is an actual new organism. So you can plant those and see how they grow. And so he figured out a lot of genetics as a result of that. And so genetics in this podcast I'll talk about are Mendelian or simple genetics. He identified the gene and he came up with 2 laws, the law of segregation and independent assortment. I'll talk about those. We'll also have some problems, so I'll have some practice problems that you can try and I'll show you how to work those out. And we'll finish with genetic disorders. The example I'll talk about is Huntington's disease, and with genetic testing it opens up this whole idea of ethics and privacy. Now, what won't I be talking about? I won't be talking about linked genes. In other words, if genes are ever on the same chromosome or on a sex chromosome or caused by multi-genes, it gets really complex. And so I'll talk about those in the next podcast on advanced genetics. But know this, that simple Mendelian genetics, the rules are simple rules of math, rules of probability. And if you understand those and how to do the first cross that I show you, you should do well in most any of the crosses you get in genetics. And so this is what Mendel did. He crossed purple flowers with white flowers, and he got purple flowers. Now, a few things that you should know. The first cross in any genetic cross is called the P cross, or the parental cross, and then the offspring of that are called the F1 or the filial one, or the offspring of that cross. And so at the time of Mendel, everybody believed in this idea of blending, that if you cross 2 parents, so mom and dad, their kids look a lot like them, and so it's maybe a blending of they didn't know that they were genes, but something inside of them. And so when he crossed purple with white and got purple flowers, that made total sense back then. This kind of fits in this blending. But what he did next was he crossed these purple flowers with themselves. And what he got was a 3-to-1 ratio of purple to white. And that white returned just as crystal white as that first white was to begin with. And so what he said is that there's a character, a trait that's passed through here, but we now identify that as a gene that's carried distinctly through each of those generations and then shows up again. Now, if you know anything about genetics, you'd know that in simple Mendelian genetics, it looks like purple is dominant, white is recessive, but we know that because of the work of Mendel. And so this is what The second cross is the one that was puzzling, and this is what he eventually figured out. So these offspring right here were hybrid for the trait. And so if we look at the parents would be big P, that represents one purple flower. Little p represents the white flower, and so each of these would be big P, little p. And so you can use a Punnett square. A Punnett square, you put the pollen or the male here, the female right here, and each of these genes get a certain column. And so this would be one parent, big P, little p, and so you simply write that across. So we got big P and little p here. We got big P and little p here, so this represents the two different parents crossing with each other. And then we simply figure out what comes. So here's big P. So we get a big P from one, a big P from the other, so that'd be big P. Since big P is dominant, we get a purple flower. On the next one, I'm going to take a big P from here, a little p from here, and so that would be still purple because this one is a dominant allele or a dominant gene. Down here, we get a big P from here and a little p from here. And so that's purple. And the reason we get white flowers is that you get a little p from both of the parents. And so the neat thing about a Punnett square, and that's what this is it not only allows you to quickly do the probability, but it shows you the percent we should get in those offspring. In other words, 3 of them should be purple, and 1 of them should be white. So we should have a 3-to-1 ratio. And if you ever get a cross like this in a problem, for example, a cross like this, just, if you're not sure what the offspring are going to be, just do a simple Punnett square where these two alleles or versions of the gene are going to be on the top and these two are going to be on the side. And we'll do some practice problems in just a second. And so what are Mendel's laws? Well, the two things that he figured out are the law 1, Mendel's law 1 is called the law of segregation, and Mendel's law 2 is the law of independent assortment. And so let's start with law 1. And so if we ever, and I got a coin here because the actual way you get genes are almost like a coin flip. So if you think about a coin, it has heads on one side and tails on the other. When you flip a coin, what are the odds that you're going to get heads or tails? Well, it's a 1 in 2 probability that you'll get heads. Same thing with genes. And so if this is that F1 generation, and this shouldn't be B, this should be P. So we'd say this is big P, little p. What are the odds that the offspring are going to get a big P? Well, it's a 1 in 2. What are the odds that they're going to get a little p? It's 1 in 2. And so that separation of those two alleles is called segregation. And so this idea of segregation says that there's a 50% chance you're going to get either of these genes. And so that's segregation. They separate, and it's just random chance. The next one is the law of independent assortment. The law of independent assortment says that this gene, the gene that causes, for example, hitchhiker's thumb, which is where your thumb actually bends back, and the gene that causes an attached earlobe, so right here I've got a free earlobe, those two traits don't affect each other. In other words, they sort independently. And so we can work problems without mixing these two together. They're going to not influence one another. Now, sometimes we'll find, for example, that some things do travel together. So you'll notice that people who have red hair also have freckles. And that's because those two genes are actually found on the same chromosome, and so they seem to travel together. And so we're not going to deal with linked genes again. We'll do that later. And so
I hope that video was able to give you a better understanding of the breakdown of genetics and how things are occurring and how we have carriers and people that are affected and people that are not affected. So to look briefly at some of these different disorders, autosomal recessive disorders is when a child inherits an autosomal recessive gene from both mother and father. Okay, it has to come from both the mother and the father to receive the recessive disorder. To see a breakdown of examples, look at your book for this class on page 13, and to look at the pedigree chart, please look on page 14. I couldn't find a good photo to do the book's photo justice, so take a look on those pages. Now for the autosomal dominant disorders, there are 4,500 different types identified, and they are caused by a single abnormal allele. So to see the pedigree chart for this, take a look at page 16 for the pedigree chart. And finally, the X-linked disorders. These genes are linked to the sex genes, so commonly the X chromosome. Primarily affects males, so the disease is passed by carrier mothers. And 25% of males and 10% of females are affected by X-linked conditions. Some examples of X-linked conditions include Fragile X syndrome, Duchenne muscular dystrophy, hemophilia, and Rett syndrome. Next, we look at revising Mendelian genetics. Genomic imprinting. According to Mendelian genetics, the phenotype of an individual should be the same whether the given gene is inherited from the mother or the father. This is not always the case. For instance, if a particular deletion occurs on the long arm of the chromosome 15 derived from the father, the child will have Prader-Willi syndrome. If the same deletion occurs on the chromosome 15 derived from the mother, the child will develop Angelman syndrome. Different depending on which parent has the deletion. Anticipation. This is the manifestation increase in severity with each subsequent generation. So the disease becomes worse with every person that contracts it. This is the case with Huntington's disease. And finally, mitochondrial inheritance. Mitochondria produce energy needed for cellular function through oxidative phosphorylation. They possess their own separate DNA. They contain genes different from those contained in nuclear DNA, and they're inherited from the mother. A mutation in mitochondrial gene can result in defective energy production and then a disease state. Finally, we'll take a look at environmental influences on heredity. Here, you mainly have your genotype and phenotype. Genotype are the particular genes that a person possesses, whereas the phenotype are the manner in which those genes are expressed. So a few examples are listed below on different types of diseases caused from the environment. And, the phenotype expresses these genes because of environmental factors. So when we get down to it, chromosomes and genetics are unbelievably complicated. And although we may simplify them by assuming tall parents will have tall children and blonde-haired parents will have blonde-haired children, a lot can go wrong anywhere along the growth and development line. I hope you're able to learn something from my presentation and that it was not too overwhelming. I understand the material is pretty complicated. I myself had to read over it quite a few times to wrap my mind around everything. But I look forward to viewing everyone's comments and everyone's Group 1 videos. Take care.